we'll call up all the speakers and we'll take a few questions for those that would like to stay. And I thank all the speakers for a very wonderful session. Okay. Are there any questions? If there's a question, please step up to the mic.
But as a molecular biologist, I would suggest we are not going to call the E6, E7, E5, E4 as a gene. Because as a gene, we consider each gene should have a promoter in the front. And also in particular for papillomavirus field, each gene should have a product for messenger RNA. But for the papillomavirus, the gene actually we consider as open reading frame. The inverted into a messenger RNA which is composed of multiple open reading frames. So I prefer in the papillomavirus field we call each gene as actually now as open reading frame. Otherwise it's very confused with the gene definition.
So you're suggesting calling them open reading frames and not genes?
No.
Yeah, no, I agree with that completely.
Thank you very much.
Any other questions or comments?
Yes, a question about deep sequencing. What qualifies sequencing as being deep? Is it that you're doing the entire plasmid that's amplified, or I think that wasn't really clear from your presentation. What's the definition of the sequence being deep?
Deep sequencing just refers to the coverage. A lot of those primate genomes that I showed you in one of my slides have only been sequenced to 1x to 2x coverage. Deep sequencing is down to 60x, 30 to 60x coverage. So what it means is that you've got 30 to 60 bits of information that overlap for that particular region. That's deep sequencing.
Okay, any other questions or comments?
Yeah, one short question. In the ecology literature, there's textbook examples of evolution being, competition between species. And in Dr. Bravo's presentation, there was a short bullet point saying that there is no interaction or little evidence of interaction between HPV species. Could you elaborate a bit on that?
So,
well, that was one of it was written as one of the dogmas of the research, or that which is currently thought, but I think there are evidences for recombination between certain papillomaviruses that has happened in, well, there are bioinformatic evidences for recombinations in some whale and dolphin papillomaviruses. There are evolutionary bioinformatic evidences also for recombination within alpha papillomaviruses that I have shown you there, specifically what has later defined what is commonly called as high-risk and low-risk papillomaviruses. There is another evidence of recombination in an experiment in the lab by Neil Christensen, where they infected a rabbit with two different plasmids, and they recovered a sequence that was a chimera of the two, so saying then or showing that may happen, a recombination within a cell in a infection. And then later there is the fourth evidence that I think there are is the retrieval in a bandicoot of a virus with features of both polyomaviruses and papillomaviruses, with the early genes of one and the late genes of the others. So I think that there are some evidences that suggest that there may be, under certain circumstances, interaction meaning recombination between papillomaviruses within a cell during an infection.
okay, let's, thank you. I think that raises a good point. And just statistically, there does not appear to be, niche, I guess, embarkment from what we know, but we're still moving forward collecting data in the post-vaccine world. Next question over there.
Yes, is there any adaptation or plans for adapting the deep sequencing to integrated papillomaviruses like in head and neck cancers or cervical cancers?
You mean looking at integrations?
Yes, of the integrations, and there may be several, but it's not clear whether an integrated genome is really a unique version in a specific cancer, or again, if you have mutations and different cell lineages.
Well, I think you raise a great question, but I think it's beyond the scope of kind of our session is really looking at the genome itself. I think the integration and the role of integration, the integrated sequences is a great issue. We'll move that to the molecular biologists. Another question.
Another question is about the HPV whole genomic sequencing. My question is, what's the lowest virus concentration for HPV whole genomic sequencing?
We haven't checked the concentration in the samples, but I think it's not too much. You don't need too much HPV DNA because we use PCR system to amplify full genome HPV16. So, yeah, it's possible, maybe even a very low level of HPV16 genome in the sample.
So do you know what the copy number would be?
Yeah.
What's the copy number? How many copies can you detect by, your technique?
Sorry, what?
How many copies?
Analysis sensitive for whole genomic sequencing.
How many viral copies? 10 copies, 100 copies, 1,000 copies?
Maybe 1,000.
1,000 copies.
Yeah.
Okay.
Thank you.
Any other questions? Yes.
Yeah, what long-term effects may vaccination have on viral evolution?
You mean at the population level?
Yeah, of course. Assuming a wide coverage of vaccination in about 50, 100 years.
Well, the thing is, to our better understanding, the papillomaviruses use the polymerases of the hosts that they infect, so ours, with a mutation rate that is very, very low, some 10 to the 8, 10 to the minus 8, 10 to the minus 9 errors. This was say this, I would not expect an evolution, as you say, in the sense of adaptation, so that there may appear new variants of HPV16 that may escape the vaccine. And also so the dynamics of papillomaviruses does not suggest that there is going to be a replacement for other types that are not targeted by the vaccine. But this is again a suggestion. There are some hints from a group in Finland saying that there may be a hint for a very, very low level of replacement for HPV33. But again, everything is very, very preliminary because we still do not have data on this sense. But then if the question is whether if we target one of the variants of HPV16, this one is going to escape, or so that there will going to be escape mutants like in hepatitis C or in HIV, then the answer is I doubt it.
Okay, thank you. Is there another question?
Yes, I have a question for Dr. Kukimoto. I was wondering, how do you consider the prospects for using universal primers for your full circle PCR?
Prospect for my study, no?
So the prospect for using universal primers for the full circle PCR to sort of capture more than HPV16, but the wider range?
Yeah. We are now analyzing other types, HPV 52 and 58, which is very popular types in Japan and East Asia. So we are now expanding our analysis to these types.
Okay, we have more questions.
Do you think the deep sequencing suggests that the evolutionary rate of papillomaviruses may turn out to be faster than anticipated?
It's a very difficult question. I have no idea whether it's related to HPV evolution, but you can see some evolution in patients for HPV variation. So I think it's very interesting to get some insights from this genetic variation from patient sample for HPV evolution. But it's not clear right now.
Okay.
I think, John, if you look at the viruses that actually come out of the patients, they tend to be a lot more stable than what, I think that the next-gen sequencing is detecting a cloud, if you will, of viruses in a patient, but I think what comes out and infects a new patient, I think is going to be as stable as we think it is. So in that sense, the evolutionary rate, I think, will be what we think it is.
Well, I think I agree with, I think that that's one of the exciting aspects, that we haven't looked at the rare mutants very much in HPV as they have in the human genome. And I think with deep sequencing, we're going to get a better idea of what the error rate, at the replication level will be. Now, whether that translates into infectious particles with transmission of these variants, still to be determined.
I think relating to an earlier question is the new selection pressure provided by vaccination might act differently than it did selection pressure previously.
Yeah, I mean, I think we're only going to know through empirical study. Another question.
How much evidence is there for cross-species transmission of papillomaviruses?
Well, we know there's some between like bovines and horses, but...
Yeah, so there will be a workshop right now at 11 on animal papillomaviruses. And so that I think we will address this question in more depth there. But BPV1 is the paradigm of a virus that can infect other species, even very distant species like not only artiodactyls but also perissodactyls, for instance, horses and zebras. We also have evidences in whales and dolphins in which we have found in individuals of different species in lesions, in genital lesions, the same viruses. And then later on, people working on the wild with these cetaceans, they told us that these species actually interbreed in the wild, so that they have sexual intercourse, meaning that there is the plausibility of this transmission exists. So meaning that in general, that I think that in most cases, the barrier to interhost transmission may be rather cultural instead of biological. And I think that may be what hinders.
Right, so there's certainly, I think, objective evidence that there does exist. It's pretty rare. What the mechanism, why it's rare, we don't know. But let's go on to the questions. Go to the back.
Robbie,
I think it's Tom.
Can I add something just to add something to that?
Okay.
There are certain expectations you might have for the depth of viral lineages with respect to the human lineage. And if the depth of those lineages is larger than what you would expect, then you might also think that perhaps humans have passed viruses across species, and the species that they would have passed them across are Neanderthals and Denisovans. Since you have evidence for hybridization or for introgression of those species, it's very possible that there's been some passage of viruses across the species boundary there too.
Okay, another question?
This might be a more theoretical question than what the methodology can provide, but how do you see, did the Neanderthals have the same HPVs as modern humans? And has anyone had an idea to look at that from the provided samples?
Well, so the problem is, yes, because we have thought about this, and the problem is they got the genome, the Neanderthal genomic information is usually from bone or from non-epithelial sources. So, and truth be told, the actual Neanderthal or Denisovan genome in the sample that they obtain is like less than 1% of the total sequence. So, some people have looked, I think we've looked a little bit and could not find any traces of papillomavirus in the sequence, but I don't think we have an optimal sequence from archaic hominids. But it's a good question.
Again, the inferences would be made based on coalescence of lineages. And if the coalescence of some of the viral lineages don't fit with what we know about the timing of movement and divergence of human populations, then the next logical explanation for that is that perhaps there was some transfer of virus prior to, transfer of the virus across species boundaries. And the culprits would be Neanderthals and Denisovans.
Are there any more questions? Okay, we'd like to thank you all for staying the extra few minutes. Enjoy the rest of the meeting.